looking at how to build a protein. So, keeping in mind that the role of DNA is just to contain the information on how to build a protein. So when we look at the function of DNA, it's really trying to use specific sections or genes to build specific proteins. And so far, we've looked at how to do the process called transcription. We don't need to use all of the DNA, we just need to use a small section. We don't want to cut that section out, so instead we create some sort of copy of that section. The machinery that we have to do that is an enzyme called RNA polymerase. We're going to build an RNA copy of that gene. Now that gene might become an enzyme that is part of a pathway. That's what Beadle and Tatum discovered, is that specific sections of DNA code for specific functional units, such as an enzyme. And then we've done some research on the molecular level. What does it look like during protein synthesis? And there are two steps during protein synthesis. The first step is called transcription. And this is when we use a gene, a DNA section, and RNA polymerase will use complementary base pairs, RNA base pairs, to build mRNA or messenger RNA. So depending on what the DNA code is, now remember, there's a coding strand and a template strand. So depending on what is coded in the template strand, the sequence of DNA letters, remember that's the key to making each gene different, is this sequence of nucleotides, specifically the sequence of nitrogen bases. So when we performed transcription, we were given a certain unit or certain section to transcribe. And RNA polymerase is going to actually do all of the steps. It's going to unzip, it's going to base pair, it's going to create mRNA. It still builds in the 5' to 3' direction. In eukaryotes, we do see there are some extra steps to make a mature mRNA. We're going to need to remove the interfering sections called introns, and we need to splice together the expressed sections called exons. We use something called a SNRP or a spliceosome to do that. We also add a 5' cap and a poly A tail. And then the mRNA will leave the nucleus, and that is transcription. The next step is called translation, and that's where we want to practice today. Translation is where we're going to see rRNA, or the ribosome, and tRNA come into play as well. So during transcription, we're making from scratch a whole mRNA. Once we get to translation, the ribosome already exists, and the tRNAs already exist, and we're just using them. So translation is where we're going to go from a sequence of nucleotides to a sequence of amino acids. So the key point in translation is the idea that we're actually going from the language of nucleotides to the language of amino acids, and that's when we will be very close to having a protein. So we know the differences between DNA and RNA. We know how to get an mRNA. And now we need to look at, well, an mRNA is not a protein. I'm not done. So, transcription produces a messenger RNA sequence. The sequence is really important. The order of nucleotides is very important. So from just 4 different nitrogen bases, we're going to come up with almost an infinite number of possible mRNAs. Some mRNAs are thousands of nucleotides long, like even 20,000 nucleotides long. Think about each of those spaces, each of those 20,000 spots could be an A, a T, a G, or a C. So we are going to see almost an infinite number of different mRNAs, and it all comes down to the sequence or order of nucleotides. Every time it's made, it should exit the nuclear pore and become attached to a ribosome to begin the next stage of protein synthesis. So we've got a lot of steps here that could go wrong. We have to make it. It has to be able to leave the nucleus, and it needs to get to the actual ribosome. And so the next stage of protein synthesis is called translation. Translation is going to occur, well, first let's talk about what it is. Translation is the assembling of amino acids. In order according to the mRNA sequence. So somehow we're going to have to get the amino acids and assemble them. So assembling means to build. So we're going to be doing some sort of enzyme action, and that's actually the ribosome that's going to be doing that. So we'll talk about the small and large subunits in just a second. The amino acids are going to have to come from somewhere. They're kind of the key. And so tRNA, or transfer RNA, is actually going to be bringing the correct amino acids. And we're going to see that this mRNA sequence is, I'm sorry, that should be is read in codons, or sets of three nucleotides, or nitrogen bases. Really, what we read are the nitrogen bases, but we do want to keep in mind that that nitrogen base is part of a nucleotide. So this process is going to be kind of the second stage, and it's when we actually get a polypeptide. We do need to keep in mind what a polypeptide is. This is our primary, if you go back to protein folding, this is the primary level of protein folding. This is just the amino acids held together by peptide bonds. So all that translation is going to do is put those amino acids in the correct sequence or order according to the mRNA. We won't have a functional protein until it does all the coiling and the folding. So the primary sequence, remember, is that first stage, and that's what we'll have at the end of translation. So we want to look at what exactly is going on in translation. So the main idea of translation is to use the mRNA code to assemble a protein out of amino acids. And this is going to occur after transcription. It's going to occur at the ribosome. And it's going to include all three types of our RNA. So we want to look at that. We know where the mRNA is coming from. The mRNA was produced and perhaps modified during transcription. The rRNA is actually going to be produced in what's called the nucleolus, or a part of the nucleus that produces rRNA, and it's going to fold with proteins to form the two subunits of the ribosome. So when you learn in introductory bio that the ribosome is basically this dot, that's not actually accurate. The ribosome has two subunits, and it's made of rRNA and protein. So it's a little bit of the chicken and egg. Which came first, the ribosome or the protein? The ribosome is a protein. But how do we make this protein if we don't have a ribosome? Chicken and the egg. So what we are going to see is that these two subunits are actually going to act as an enzyme. They are going to help form the peptide bonds. tRNA is a very important molecule that when you are in introductory bio, you probably remember it looking something like this, like a fork. And we'll talk about the important parts, but it turns out that it is just RNA. But this RNA actually pretty unique. It is going to fold and base pair on itself to make what we call a clover leaf structure. This is a 3D rendering. You can actually see the nitrogen bases hanging off of the sugar phosphate backbone. The job of tRNA. So again, we are a single strand. All three of these are single-stranded, but they'll fold and base pair on itself. tRNA is going to transfer or deliver amino acids. So the amino acids are going to be connected up here. Of course, we know what an amino acid looks like. It has that central carbon, another carbon over here, a nitrogen. Here we have an double bonded to an O, bonded to an OH, an H, and an R group. So an amino acid will be hanging out up there. And then we're going to have an active part down here called the anticodon. And the anticodon is going to help us do this transfer or delivery by using the anticodon sequence, 3 nitrogen bases, to be attracted to an mRNA codon. So it turns out we're going to use base pairing in translation as well. The anticodon that's here will be attracted to a codon on the mRNA. So this diagram is really helpful because what it's going to show us is kind of the steps of translation. So the first step is initiation, and that is actually going to include first the mRNA binding to the small subunit, then an anticodon, and then the large subunit. So initiation here actually has 3 steps. The mRNA binds to the small subunit, a tRNA is attracted, and the large subunit comes in next. It turns out that the start is always, the first mRNA is AUG, which means that the first anticodon will base pair to this. RNA base pairing to AUG would be AUG, C. So this is actually just how translation always starts. The small subunit always binds so that a tRNA is always attracted, and then the large subunit will bind. One thing to notice is that the large subunit has 3 active sites, the A, P, and E site, or the EPA. The E is for exit, the P is for peptide transfer, and I'm not sure why they named the A. But what we're going to see here is that as other tRNAs come in, we're actually going to form a bond between the amino acids. So we do want to be able to actually go from start to finish to a functional protein. So we're going to practice that a little bit. So how does a sequence of nucleotides determine a sequence of amino acids? So a codon is 3 letters on mRNA, and you're usually going to be given an mRNA codon chart, and that will give you the amino acid. And so triplet is 3 letters. And so we use this chart, which you should be able to follow, to find the amino acid. But the anticodon is 3 on tRNA that brings the amino acid. So we'll practice this a little bit more, but the main idea is that you can use an mRNA sequence to find the amino acids, but also that you can describe everything that's going on. So that's where we'll stop.